Hi. Okay, so it's my pleasure to introduce our speaker today. Dr. David Knowles is a postdoctoral researcher working with Jonathan Pritchard in the genetics department and Sylvia Plevridis in the radiology department at Stanford University. Prior to that, he received his PhD in machine learning from the University of Cambridge, working with Jovin Garamani, focused on Bayesian non-parametric models and inference. His current research lies at the most exciting intersection of machine learning and genetics, addressing very important basic questions of how DNA mutations lead to diseases. And his research has been featured in very prestigious biological journals, such as Nature Methods, Nature Genetics, and Science. And part of his machine learning research during his PhD won the Lindley Prize, a prestigious award from the International Society of Bayesian Analysis. And today, he's going to talk about the probabilistic models of gene splicing, which can tell us about the underlying mechanisms of many human diseases. Please join me in welcoming Dr. Knowles.
Thank you, Suren, for that kind introduction. So as Suren said, I did my graduate studies in statistical machine learning, and one of the areas I focused on was this idea of Bayesian non-parametrics. So with Emily Fox being here, probably many of you are aware of this class of methods, but for those who are not, these are algorithms which try to adapt their model capacity to the data at hand. So kind of the canonical example would be if you're doing a cluster analysis or trying to fit a mixture model, then trying to, from the data, get a sense of what's an appropriate number of clusters for the data. An example that's closer to my heart is sparse factor analysis, which would be, you could think of as trying to learn from the data what's an appropriate number of sparse principal components. I also worked on Bayesian non-parametric methods for spatial temporal data, as well as in network analysis and Bayesian hierarchical clustering. Another class of methods I worked on are what are known as variational inference methods. So these are deterministic alternatives to Markov chain Monte Carlo, which would be kind of the default way of doing inference in Bayesian probabilistic models. And the work I did in this area was had the aim of extending these class of methods to a broader range of probabilistic models. During my PhD, I got increasingly interested in applications to genetics and genomics. So one of the first projects I collaborated on in this area was looking at epigenomic changes during the development of cardiac disease, and also looking at changes in gene expression with age in different tissues in a cohort of twins. More recently, I've led projects in genomics, and two that I want to mention that I unfortunately won't have time to talk about today. One is looking at detecting gene-by-environment interaction effects on the transcriptome using allele-specific expression, which is something that you can get from RNA sequencing data, which you couldn't get from microarrays. And most recently, looking at the genetic basis of cardiac side effects during chemotherapy. And this is joint work with Yoav Gilad's lab, and we have a preprint up on bioRxiv if you're interested in looking at that. So I want to sort of motivate for the more computationally minded people in the room why it's a really exciting time as a computational scientist to be thinking about problems in biology. And a big motivation for that is that the scale of data being generated in genomics is really exploding. So this is a figure from a 2015 paper in PLOS Biology, where they looked at the cumulative number of human genomes having been sequenced over the last 2 decades. And in case you can't see it at the back, the y-axis here is on a logarithmic scale. So this growth is quite clearly exponential, and so the only real question is what's the doubling rate here. So this paper estimates that the historical growth rate recently is a doubling every 7 months, Illumina, which is the company that makes most of the sequencing machines that most of the sequencing is done on, put that estimate at a year. And either way, this is outstripping Moore's law. And someone recently pointed out to me that if you extrapolate this on to about around 2025, then it would suggest every human in this world will have been sequenced, which is maybe not realistic. But the ability of modern DNA sequencing technology has really evolved in leaps and bounds over the last decade. The application that I'm really interested in using this new data for is to understand this process called alternative splicing. And for those of you who don't come from a biology background, I want to give a little primer on what this process involves. So probably most of you are aware that DNA is the long-term information store of the cell. It's made up of these nucleotides, A, G, C, and T. And to give you some context, the human genome contains around 3 billion of these base pairs, or nucleotides. The genome is made up of important functional units, most important of all, genes. But in higher organisms like humans, genes themselves consist of these regions called exons and regions called introns. And for now, you can think of these introns as junk DNA regions that have to be removed before we can actually make the protein that we want. DNA, through a process called transcription, is copied to what's called precursor RNA. So this is going to act as the intermediate before we make protein. And splicing is the process by which these intronic regions are removed to give what's called a mature messenger RNA. That messenger RNA is then translated into a protein. And the proteins are what we think of as being the functional units of the cell. So these molecules can act as things like structural proteins or enzymes. Alternative splicing is the phenomenon that can be variation in which regions are removed as introns. So what I'm showing here is the production of a different messenger RNA, known as a different isoform, through alternative splicing, where in this case, this middle exon 2 is included in isoform 1, but excluded in isoform 2. And this will result in the production of a different protein. To motivate why this is an interesting phenomenon to study, I want to introduce you to the tau protein. So tau is a protein whose job is to stabilize structures called microtubules. Microtubules are particularly important in neurons, for example, in the brain. And neurons have these extensions called axons, which you can think of as the output channel of a neuron. The extensions here are known as dendrites, so these would be the input channels. Axons tend to be really long compared to the cell body. So an axon might be 1,000 microns long, whereas a cell body is typically only up to around 100 microns. And, this makes it really challenging for the cell to transport the required nutrients and proteins up and down this long distance. And so what you can think of the microtubules as a railway network. These allow the cell to transport the required molecules up and down these structures using these proteins called kinesins. The tau protein itself is transcribed from a gene that's made up of 14 exons. And this is pretty typical for a human gene. The average is around 9. There's various alternative splicing that happens in tau, but I want to focus on exon 10. And you'll see the reason why in a second. So there are two major isoforms of tau that are created by alternative splicing, 3R and 4R. The reason they're called this is that the 3R version, which excludes exon 10, contains 3 microtubule binding domains, whereas the 4R isoform contains 4 microtubule binding domains. As you might imagine, if you have more microtubule binding domains, you end up with higher microtubule binding affinity. And this effect is actually even bigger than you might think, because it turns out that exon 10 actually encodes one of the strongest microtubule binding domains. In healthy development, in the fetal developing brain, it turns out you see almost exclusive expression of the 3R isoform and almost no expression of 4R. Whereas in healthy adult brain, you see an almost 1-to-1 ratio of the two isoforms. So this is an example where in healthy development, there's variation in the splicing choices that are required for healthy neuron function. It turns out that in many diseases of the nervous system, for example, frontal temporal dementia with Parkinsonianism, there's an observed imbalance between these two isoforms. And while in this particular example, you see greater expression of the 4R isoform compared to the 3R, there are also examples, including Alzheimer's, where the ratio is the other way around. So this suggests that splicing, RNA splicing, is an interesting process to study, both in terms of healthy development as well as its role in human disease. To try and understand and query splicing in different samples, a modern technology that's becoming ubiquitous is RNA sequencing. So I want to give you a brief primer of what RNA sequencing is. So RNA sequencing starts out with purified messenger RNA molecules. These are then fragmented into shorter segments and reverse transcribed. So this is creating a DNA copy of the original RNA molecules. The reason we do this is that these, what are known as cDNA or complementary DNA fragments, are something that we can put through a next-generation sequencer. DNA is the thing that we're currently really good at sequencing, and we're not very good at sequencing RNA. So we can sequence these fragments, and typically on modern Illumina machines, you might get read lengths of, say, 75 to 100 base pairs. So those resulting short reads, we then need to align to the genome, and I'm showing that just for one small gene with 3 exons here. So some of these reads are aligning to a single exon, but I want to also draw your attention to what I'm calling spliced reads. So these are reads where a portion of the gene maps to one exon, but the remainder maps to a second exon. And these reads are going to be really valuable for determining what splicing choices were made by the cell. So the projects I'm going to talk about today both use RNA sequencing to try and better understand splicing variation and its role in human disease. The first is an algorithm we developed called Leafcutter, which quantifies splicing variation from short-read RNA-seq, and this was published last year in Nature Genetics. And then I'll talk about ongoing work trying to better understand the genetic basis of splicing dysregulation. So before I talk to you about our methods of splicing quantification, I need to tell you broadly what's been done in the past. So existing methods for splicing quantification fall into 2 main categories. The first are broadly known as isoform ratio estimation methods. So these use a known set of gene isoforms. And this example I'm showing here is taken from a nice review paper by Leo Pachter from 2011. And what he's showing is 3 different isoforms here for the same gene and corresponding short RNA-seq reads, as well as the ambiguity in the assignment of these reads to these different isoforms. So this first read could have come from any of these 3 isoforms. The second read must have come from the blue or the green. And actually, only this one splice read, D, unambiguously gets mapped to the red isoform. There's been some nice work trying to view this as an inference problem, where you view the isoform expression levels as latent variables and then try to jointly estimate the isoform abundances as well as the assignment of each of these reads to different isoforms. However, especially for large genes with many different isoforms, this is a really statistically challenging problem, and the resulting estimates tend to be noisy and tend not to be robust to small changes in the input data. In addition, you're also very dependent on having a good set of known isoforms to begin with. The second set of methods try to use the idea of focusing on a specific exon, say this exon in the middle here, that's bordered by two what are known as constitutive exons. So these are exons that are always, we think of as always being included in the gene. And the percent spliced in will be the proportion of the time that this focal exon is included in the gene relative to its neighbors. So if the RNA sequencing reads mapping to this region looks something like this. So I'm showing 3 reads here that map to the focal exon, and then these representing spliced reads that map over the over 2 exons. Then 7 of these reads that I'm showing unambiguously support inclusion of this exon in the transcript, whereas 3 of these reads unambiguously support exclusion. And I'm not going to go through the math here, but you can, accounting for read length and exon length, you can use these numbers to estimate the proportion of the time this exon was included. The problem with this method is that while it does a good job of quantifying simple exon skipping events, where this exon sometimes is and sometimes is not included, there are many other splicing events that it's not able to model well. So one, for example, is 3' or 5' extensions. So I should say that the 5' end of the sequence is upstream and 3' is downstream. So this, for example, would be known as a 3' extension of this exon. The exonic percent spliced in also can't really model when the situation when multiple exons are skipped, when there are mutually exclusive exons, or complex splicing events where there's a combination, for example here, a combination of exon skipping and a 3' extension. So our solution to, or our approach is to take a third way, which is to think about, rather than quantifying exons, to quantify intron usage rates. And this sounds like a really simple change, and in some ways it is, but it comes with a number of computational and conceptual advantages. So the first is that introns are uniquely defined by spliced reads. So if you give me a spliced read, I can tell you exactly the start and end of the intron that it supports. Or you can think of this as being a one-to-one mapping between spliced reads and introns. That one-to-one correspondence means that quantification is very straightforward. To say how much an intron is being used, I just have to count how many spliced reads correspond to that intron. This also makes us much less reliant on having accurate known gene models, because the data itself, the spliced reads, define the units that we're going to quantify. And unlike the exon percent spliced in, we're also able to model the complex splicing events that I showed you before. So there is a remaining question, which is, what's our unit of quantification going to be? Or you can also think of this as, how are we going to normalize the counts that support the use of each intron? So if we just looked at the counts for an intron, our signal would be completely confounded by total expression for the gene. And since we're wanting to quantify splicing, not expression, we need something to normalize by. And we came up with the concept of splicing components, and I think this is most easily shown graphically. So consider this gene that has 3 constitutive exons, as well as an alternative exon, and this 5' extension on this final exon. The spliced reads that you might see in this region might look something like this. And the way we just define our splicing components is as follows. So consider a graph where the nodes in the graph correspond to starts and ends of introns, or starts and ends of spliced reads equivalently. And we draw edges in this graph whenever we observe a spliced read between 2 of these nodes. The splicing components that we use are then just the connected components in this graph. So in this case, we'd get 2 splicing components, one corresponding to the left side of this constitutive exon and one corresponding to the right-hand side. the quantification that we're going to then think about is the relative usage of each intron relative to the other introns in the same splicing component. And I just want to emphasize here that this lets us model all of those more complex splicing events that I showed you a couple of slides back. And I'm also showing here the spliced reads that support different splicing choices in each of these settings. The first thing we wanted to use these quantifications to do was to look for differences in splicing between different conditions, or in particular, in this example, I'm showing between different tissues. So here I'm showing data, RNA sequencing data from brain and heart samples for this an important gene called RBFOX1. And the visualization here is showing the average proportion of the time each of these introns is used relative to the other introns in the splicing component. And the thickness of the lines corresponds to that number also. And what you can see here is that in brain, there's preferential use of this first exon, whereas in heart, there's preferential use of this exon. But we want to be able to statistically test for such differences. And we do this using a Dirichlet multinomial generalized linear model that directly models the observed counts for all of the introns in our data set. I'm not going to go into this model in detail, except to say that the multinomial likelihood corresponds to the fact that we're thinking about the choice of which introns were removed relative to the others in the cluster. The Dirichlet component lets us model overdispersion in RNA-seq data. If you've seen beta binomial models be used, and which also get used sometimes in genomics, then this is just the multivariate generalization of that likelihood. The linear model component means that we can easily include both the covariate of interest, which in this case would be an indicator variable saying which tissue the sample is from, as well as technical confounders like sequencing batch. We optimize the parameters of this model using LBFGS in Stan, which is a probabilistic programming language developed at Columbia. And we do hypothesis testing using likelihood ratio tests. So we find that the likelihood ratios are well approximated by a chi-square distribution asymptotically. We initially tested this methodology out using the data from the GTEx consortium. So this is a large NIH-funded RNA sequencing project where they did RNA sequencing on 44 different tissues in over 400 donors. And the data is publicly available. And we used Leafcutter to look for differential intron usage patterns across GTEx tissues. We find thousands of differentially excised introns between pairs of tissues. And what I'm showing here is just a heat map for the 500 most differentially spliced introns between pairs of tissues, and then a hierarchical clustering. And so I should say rows here correspond to introns, and columns correspond to individual samples from the data set. And what you see broadly is that the different tissues cluster together when you use this quantification. And this is in contrast to using isoform ratio estimation, where you don't see such a clear clustering. We've also looked at whether these differentially spliced introns that we see in between pairs of tissues in human, whether those differences are conserved when we look across mammalian species. And indeed, generally we see that around, for example, going as far as mouse, we find that around 10% of the differential splicing events that we find are actually conserved across those millions of years of evolution, suggesting that they do play a functional role. I wanted to show this example because, partly because we were happy that they used our method, but partly because it's also a nice little vignette about the advantages of open science. So we put the leafcutter manuscript and code on bioRxiv and GitHub respectively in sort of mid-2016. And then this paper showed up on bioRxiv towards the end of 2016. And that could never have happened were it not for bioRxiv and people putting code on GitHub. So what this group from Germany is interested in is looking at trying to genetically diagnose rare mitochondrial diseases. So they took 100 patients who they suspected of having a mitochondrial disorder, and all 105 had exome sequencing. So this is DNA sequencing of just the exons. But 48 of them had received no genetic diagnosis. So there was no mutation at the DNA level that in a gene that made sense for their condition. So what this group did was to take fibroblasts from these patients, from blood, and do RNA sequencing. And then they looked at 3 different things. So they looked at aberrant expression, so expression outliers. They looked at aberrant splicing using leafcutter. And they also looked at monoallelic expression, which I'm not going to go into. They also did some nice functional and biochemical validation of the dysregulated events that they found. And they were able to confidently diagnose an additional 5 out of the 48 patients, as well as giving putative diagnoses for another 23. To show you one example of this, this is this CLPP is a mitochondrial endopeptidase, which they found using leafcutter that these are healthy individuals, you see the exon skipping of this exon 5 in the gene is extremely rare. In this one individual from their cohort, you see quite frequent skipping of this exon, as well as an occasional 3' extension. And it turns out that this individual has a rare genetic variant at the end of this exon. Something else that we and others have used the leafcutter quantifications for is looking at what are known as splicing quantitative trait loci. So these are genetic variants that are associated with a splicing outcome. So I'm showing an example here from a paper from Dan Gaffney's group at the Wellcome Trust Sanger Institute in the UK, where they used leafcutter to look for associations between genotype and splicing outcomes in iPSC-derived sensory neurons from just over 100 donors. This is one example where they found that this single nucleotide polymorphism, so this is a position in the genome where there's a difference between different individuals, is associated with splicing outcomes in the CYP1L2 gene. So what they found here is that the human reference genome in this position has a C. And when you have C, you see that this exon is usually included in the gene about half the time. For heterozygotes, so individuals who have 1 copy of C and 1 copy of T at this position, you see more frequent skipping of this exon. And in fact, for individuals where they have they're what's called homozygotes, so they only have T in this position, you see almost complete skipping of the exon. And it actually turns out that this SNP, the single nucleotide polymorphism, is also associated with Parkinson's disease. We've looked more broadly at the ability of leafcutter to map these splicing QTLs, and we find that we're able to detect considerably more of these associations between genotype and splicing outcome than alternative methods. And we attribute this to both our ability to provide a less noisy quantification than you get with isoform ratio estimation, and our ability to quantify more splicing events than you can with exon percent spliced in. Our success and the success of others in mapping splicing QTLs encouraged us to try using the leaf-cutter quantifications in what are known as transcriptome-wide association studies. So the idea of what's known as a TWAS is that if you have some data set where you have genotype information, so single nucleotide polymorphisms, as well as some trait of interest, say whether you do or don't have a disease, but you have no molecular phenotyping. But then you also have these great reference cohorts, such as the GTEx data set that I already talked about, where you have gene expression measured using RNA-seq, and you also have genotyping for these individuals, for hundreds of individuals. Then you can learn a predictive model of gene expression from genotype, and then use that predictive model to impute gene expression values in your larger GWAS cohort. Sorry, I should have said GWAS stands for Genome-Wide Association Study. It turns out as long as you're using linear predictive models, you can get away with using GWAS summary statistics. So these are typically just effectively pairwise correlations between the single nucleotide polymorphisms and the trait of interest. And while people have usually only considered total gene expression as a phenotype here, we thought, well, what about also looking at splicing? So I want to show you an example of doing this for Parkinson's disease. So here we mapped splicing QTLs in prefrontal cortex samples. And we used one of the largest publicly available Parkinson's GWAS data sets. And doing this, we found 18 and 26 genes, respectively, associated to this disease through expression and splicing. So we actually found more genes through splicing variation than we did through expression. And part of the reason I wanted to show you this example is, so the y-axis, this is a genomic position, and the y-axis here is a z-score from the TWAS. And the most significant result is actually for MAPT, which is microtubule-associated protein tau, so the protein that I told you about at the beginning. So it actually turns out that it's not exon 10. It seems like it's exon 3. So the most strongly associated genetic variant with both splicing outcomes and Parkinson's disease for MAPT is this SNP in the intron between exon 1 and exon 2. And what we find is that the genotype here is correlated with the inclusion level of exon 3, and also extremely strongly correlated with Parkinson's disease risk. Something that's really cool about this is it actually lets you make a statement about the direction of effect as well. So we can conclude from this finding that inclusion of exon 3 in the gene appears to be protective against Parkinson's disease.
Do you have a mechanistic explanation? I mean, why does SNP?
No, we don't. So that would be really cool to get to. And of course, there can be longer range interactions that between that microtubule binding domains, but there are other domains that are important in the tau protein beyond just the microtubule binding domain. So yeah, you'd have to look at the actual protein structure, I think, to understand why this region in particular is important. And that's a follow-up that we would like to do. So the mitochondrial genetic diagnosis example I showed you, as well as our own work looking at Parkinson's disease, is suggestive that RNA splicing is important both in rare and common human disease. So we wanted to try to get a bit closer to understanding how genotype can influence splicing outcomes. And to do this, we built what we think of as a predictive model of splicing choices. We're calling this algorithm Grasshopper, and it models splicing outcomes as a series of competitive choices between splice sites. So what this looks like in cartoon form is consider a given anchor, or I'm going to call an anchor 5' splice site, and we're going to try and ask which downstream position is going to get used as the corresponding end of the intron. And it turns out that 3' splice sites at the end of the intron are almost always, in 99% of cases, correspond to an AG dinucleotide. So we're going to consider all AGs within 100 kilobases downstream of our anchor 5' splice site. And that's because 99% of introns are shorter than 100 kb. And a genomic window around each AG, in the end, we use 800 base pairs, will be fed into a neural network which tries to output a number corresponding to the probability of splicing at this given position. And as well as the genomic window, we also give the neural network a feature corresponding to the intron length, so the distance from this 5' splice site to the specific AG. We also learn an analogous model the other way around. So condition on a specific 3' splice site, which 5' would get used. And we use a very similar quantification strategy as we use for leafcutter. So consider a specific 5' splice site here, which has a downstream alternative exon, as well as a constitutive exon with a 5' extension. Then the different splice site choices that are actually observed are represented here. And the corresponding RNA sequencing reads supporting the use of each of these 3' splice sites are shown here. But bear in mind, there'll also be lots of AGs in the introns and potentially also in the exons that are potential splice sites that the model will consider. And those will correspond to zeros in our RNA-seq counts. We again use a Dirichlet multinomial likelihood, and the outputs of those little neural nets that I showed you are put through a softmax to give a probability distribution over the possible 3' splice sites. C here corresponds to an overdispersion parameter, and we actually fix this, and I can talk later if you're interested in how we set that. The neural network that we use is designed specifically to take account of known splicing biology. So in particular, we use a dense neural network on 80 base pairs around the AG. And this is because we know that there are specific base pairs that are particularly important in that area, and that we shouldn't have translational invariance there. Like the position relative to the AG is very important. But then we also use an 800 base pair window that's put into a convolutional neural network component because there are these elements known as splicing regulatory elements where RNA binding proteins can bind in these positions and influence whether this position is used as a splice site or not. So we use these two components that are combined through a couple more layers of fully connected neural nets to give a final splice site strength. The performance quantification we use is as follows. So we trained on all odd-numbered chromosomes and test on all even-numbered chromosomes. And here, the data I'm showing is both training and test on brain RNA-seq from GTEx. The performance metric I'm using here is for a given 5' splice site, across 5' splice sites in my test set, what proportion of the time do I correctly predict the correct 3' splice site? So the splice site that's used most frequently according to the RNA sequencing data. So this is, there's obviously other metrics you could think of, which would better account for positions where you actually do see alternative splice site usage. But this is appealing in its simplicity. So bear in mind that when looking 100 kb downstream of a 5' splice site, there'll be typically around 3,000 AGs. So if you guess at random, the proportion of the time you'll get the right answer is around 1/3,000. So the fact that this work from Chris Burge's group at MIT from 2004 gets the right answer about 10% of the time is actually pretty impressive. The best dense neural network we could come up with gets the right answer about 20% of the time. The best convolutional neural network gets the right answer just over 30% of the time. And our combined hybrid dense and convolutional neural network gets the right answer about 70% of the time. If we don't use overdispersion, so we use a multinomial likelihood instead of the Dirichlet likelihood, we lose some performance from not taking into account the noise in the RNA sequencing data as well. And interestingly, if we don't use intron length, we also lose a lot of performance. So this shows intron length is an important determinant of splicing choices. Something that's also interesting is that if we include what's called a conservation track from an algorithm called PhyloP, we can do even better. So these conservation tracks tell you something about how conserved each position is across vertebrate species. So positions that are important functionally will tend to not have differences between species. So this information is able to tell the model a bit about which bases are likely important for functional reasons in the cell. But of course, the cell doesn't get to look at this. The cell doesn't get to look at, is this base pair the same in human, where I currently am, as in mouse? So in a way, this is cheating. So the results I'm going to show you don't include using conservation. We wanted to understand what features the model is using to make these predictions. And perhaps the most straightforward way to do this is to use a process called in silico mutagenesis. So what this does is to look at a specific 3' splice site, and then to, in silico, ask to mutate each position to the other 3 possible bases, to nucleotides, sorry, and then to ask what the change in the predicted splice site strength is. So what I'm showing here are those changes. And so, for example, if the letter here is above the x-axis, that means that changing to that nucleotide increases the predicted splice site strength by that amount. And if it's below, then it's changing to that nucleotide corresponds to a decrease. What's really cool about this is that we find that the model's actually able to pick up on these known features of splicing biology. So there's a required component for splicing called a branch point, a polypyridamine tract. Also, the 3' splice site choice is dependent actually on having a good downstream 5' splice site so that you can make an exon. And I should point out here that the 5' splice site, that the 3' splice site it's being chosen for, is somewhere off way to the left here. Yeah. We've done a similar analysis where we've aggregated across all test 3' splice sites using a k-mer-based analysis, in this case using 5-mers. So here we run in silico mutagenesis and then ask what's the contribution of each k-mer at each position, and then aggregate that across the test set. And this recapitulates some of the features I showed you on the previous slide. So in particular, this green here corresponds to the branch point. The purple here corresponds to the polypyridamine tract. The red corresponds to the 5' splice site. But what's even more exciting here is that we actually find this GAAGA motif, which corresponds to what's known as the exonic splicing enhancer. So this is a motif that if the cell observes it in an exon, it encourages inclusion of that exon in the final transcript. We were then interested in getting to really using this predictive model to try and understand the effects of rare or de novo genetic variants or mutations on splicing outcomes. And we originally did this in the GTEx cohort since they have whole genome sequencing for about 150 of the individuals. So this is showing an example where there's a rare single nucleotide polymorphism, which exists in about 0.5% of Europeans, and it changes this G to a T just one base pair upstream of this AG. And this makes this AG into a much stronger potential splice site. And we actually see that in the one individual that has this variant that is heterozygote for it, we see approximately 50% of transcripts actually use this AG, suggesting that basically whenever you have this variant, you see splicing to this position. But what we were really excited about doing was applying this methodology in actual disease cases. And here we focus particularly on de novo mutations. So these are mutations that the disease case has that his or her parents do not have. And we've looked at this in 2 sequencing cohorts with trios. So this means the sequencing has been done of both what's called the proband, so the individual with the disease and the parents. And the first cohort we looked at was looking at intellectual disability in just over 4,000 cases, as well as in autism. And here we used 2 different cohorts with a total number of cases is just under 4,000. And we use control healthy samples as unaffected siblings from this consortium. So the analysis we did here was to use Grasshopper to categorize both intronic and synonymous. So these are mutations that don't change the protein coding sequence, de novo mutations, as whether they're splice disrupting or not. And then we compare the cases, so the individuals with the disease, to our control set, where we account for different de novo mutation ascertainment rates, which can result from different sequencing coverage. And we model the mutation rate as being, from parents to child, as being dependent on a trinucleotide sequence context. So what we find is that in our these cohorts, individuals with intellectual disability have, on average, 1.54 times more predicted splice-disrupting variants than the healthy controls. And that number for autism is just under 1.4. Both of these enrichments are statistically significant. And by doing a similar analysis for protein truncating variants, we estimate that the proportion of pathogenic, so disease-causing mutations that act through splicing is 7% and 16%, respectively. We can also use our predicted splice-disrupting mutations to improve what are known as burden tests. So these are statistical tests that try to ask if there's a statistically significant enrichment in the number of mutations observed in a specific gene in the cases versus the controls. And that helps us to detect 5 additional genes associated with intellectual disability and 2 additional genes associated with autism. Finally, we wanted to try and validate some of these predictions. And we did this by obtaining lymphoblastoid cell lines from 28 of the individuals from the Simons collection, where we had predictions that there was a de novo mutation disrupting splicing, and where there's also expression of that gene in LCLs. I'm showing one example here, this MED13 gene, which is broadly involved in transcription. And in healthy individuals, this gene, this exon, is almost always included. And there's basically never this 3' extension. What you see in one individual in the cohort is that there's a mutation here from A to G, just one base pair downstream of the canonical, the normal healthy GT, which is the usual 5' splice site. And this results in both quite frequent skipping of this exon, as well as use of this GT instead of the normal GT. Overall, we find that 21 out of 28 of our predicted splice dysregulating mutations validate in that we actually do observe in the RNA-seq data the effect we predicted. And the 7 that didn't validate, one possibility is just that these are real false positives. The other is that the model and the autism disease we expect to be acting through neuronal tissue. And then these lymphoblastoid cell lines are not really the right cell type to be looking in. So to wrap up, I wanted to talk a little bit about future directions. So I'm interested in having a lab that both develops computational methods that are able to address important questions in both genomics and genetics, and also in having biological questions drive new computational research. While I'm excited about having these biological questions come from collaborators, there are specific questions that I'm interested in addressing in the realm of RNA splicing. So one is trying to learn condition and tissue-specific splicing models. So the neural network I've shown you is pretty good at predicting splicing outcomes in a relatively tissue-agnostic way, but predicting subtle differences between tissues is very challenging. And there are data resources that can be leveraged there, including genomic methods that measure the position of the binding of different RNA binding proteins to RNA. There are starting to be data sets where it's possible to look at splicing variation at the single cell level. And this is something that comes with significant technical challenges, both in terms of modeling the noise distribution, as well as thinking about how to look at differences across multiple samples, for example, between case and control. And I'm also interested in pushing this research closer to the clinical and translational aspects. So how to prioritize the different variants that we detect in clinical sequencing studies, like the examples I showed you at the end, particularly with regard to splicing mutations. But there's also, I think, interesting work to do for the machine learning community for better automating interpretation of the outcomes of those studies. Yeah, and finally, I'd like to acknowledge the Pritchard Lab that's been my primary home for the last few years. Sylvia Plavritis in radiology at Stanford, as well as, so the LCL sequencing I showed you at the end was with Kyle Farr at Illumina, as well as many others who have been supportive through my PhD and postdoc. Okay, thank you.
Any questions?
You mentioned that there's differential expression or differential splicing based on tissue type.
Yeah.
But the genetic code remains the same based on tissue type. Do you have any sense of what drives the differential splicing in different tissue types?
Yeah, that's a great question. So there are some known examples. Actually, the RBFOX1 gene that I showed you is one of the best known examples where that's actually what's called a splicing factor, which its expression is variable across tissues. And its binding to precursor messenger RNA helps determine different splicing outcomes, depending on whether it is or is not expressed in that tissue. So generally, that's kind of the canonical way we think of it, is that there are secondary. So in this version of the talk, I don't get to talk much about the splicing machinery generally, but the fundamental unit performing splicing is called the spliceosome. And that's a set of both RNAs and proteins, but there are also all these many secondary proteins called splice factors that bind to the RNA and influence splice site choice and whether specific exons are or aren't included. And the expression of those splice factors varies across tissues. RBFOX1 is just one example of those. So I think actually something that would be, that sort of plays into the future research I talked about. So for example, trying to predict tissue-specific splicing outcomes, one thing that would be really interesting would be whether including the expression level of the known splice factors helps you predict better across tissues, because you know something about which splice factors are actually active. The only issue with that approach is that the thing you can get easily is just the RNA expression level of the splice factors, whereas really you'd want protein expression level, and then there can also be modifications that change how active proteins are, for example, phosphorylation. But yeah, it's a great question. Sorry, the other thing I was going to say was, it's something that would be also interesting to look at for the single-cell splicing, because if there's variation between individual cells in splice factor levels, that you'd expect that to result in coordinated changes in splicing outcomes across the genome for a single cell. And so that's something that we're also sort of thinking about how to look at.
Thank you.
Yeah, at least in that last example, the variant that you looked at was very close to the
Yes.
AG. But you're saying you're looking at these windows of like 400 nucleotides around the splice site. So is any variant within this window something you make a prediction about? Or how
I think the furthest one, the one that was the furthest away that we looked at was like, I want to say, I think it was around 60 base pairs into the intron. We didn't look at things further away. The strongest effects, so the model keeps doing better as you extend that window. 800 base pairs, honestly, is sort of a trade-off between, it's still pretty reasonable to train in terms of computational cost. You still see a bit of improvement if you go past the 800 base pairs. But in terms of the variants that the model predicts as having big effects, then they're never that far away. Like whenever we rank them.
When you extend the model, it actually does better in variant prediction, or just?
No, no. Well, just the prediction to held-out chromosomes, which is a very different problem. Right?
And I guess, are you worried you're just doing gene prediction, like learning?
Yes, I'm worried. Yeah, which actually I was talking to Gary about this earlier, that like it's part of my concern is it is one of our concerns, and especially once you include the phylo-P conservation track, right? Because if you look at the conservation track, a lot of it's just like, here is an exon, this thing is conserved, and then if it gets to look at that. So I think that is why you have to also look at the variant prediction and using the kind of data that you generate in parallel with looking at this natural variation, I think is probably, in the end, going to be the best way to go. Yeah.
Alex?
So about that splicing machinery, this is very similar to the first question. But you have this method for finding this flexible mapping from the sequence of like around some splice site to whether or not it's going to get spliced.
Yeah.
And one interesting question is, well, what mechanistically is governing that mapping?
Yeah.
Right? And I don't know if the data for this are out there, but I think from a genetics perspective, one cool thing to try might be if you found a lot of data where you can acquire that map for cases where the splicing machinery has been mutated.
Yeah. Which does happen in like some leukemias, for example. There are examples in cancer where the mutations are actually in splice factors, right? So, yeah.
And if you could ask how that mapping changes, given a certain mutation, is that a thing that you would be interested in doing?
Yes, I think it would be very interesting to do. I was meant to repeat questions, I'm just remembering. So the question is that what I'm doing is learning a mapping from sequence to splicing outcomes, and that mapping might actually change if you have mutations, for example, in splice factors, right? And that is actually something that's observed in some leukemias. And actually, we have a project with a group at Stanford looking at spinal muscular atrophy, where it's mutations in the SMA gene that result in pathology, and there are known, SMA is broadly an RNA processing protein. It's involved in more than just splicing, but one thing it's involved in splicing. So there we're looking at trying to understand what are the downstream effects of the SMA, of that protein being mutated. What's interesting there is that SMA is expressed pretty ubiquitously, but when you have a mutation in SMA, it's motor neurons that die primarily. And so it's sort of trying to understand why that happens. And also trying to understand if there's a small set of genes, of downstream genes that are mediating the pathogenic effect, or whether it's just broadly that splicing is disrupted. Yeah, and then cancer is the sort of obvious place to look at this, partly because there's so much data, right? So you could look at TCGA, they have RNA sequencing, they have exome sequencing, and for a lot of them, they have, a lot of the samples, they have whole genome sequencing as well. So that's somewhere where you could explicitly look for trying to associate changes in the this map, as you say, the mapping from sequence to splicing outcomes, and associate that with what mutations you see in splice factors. I mean, you could look more broadly, but I think it would make sense to primarily focus on splice factors. Yeah. It's a good question.
Okay, any more questions?
Just
one
last quick question. So when you say that in this last example, you sort of had this validation where you had, I think it was like I forget, like 20-ish so cell lines that you looked at. Like how did you validate? Is it like it's like usage?
It's just the right. It's just the right direction of effect, and then So, and we also say validation, right? I mean, it's validating the predictions. It's really not validating that they have a causal role in the disease. So I think as if you're being a real biologist, you could say that you'd want validation would be saying that if you CRISPR that variant, then some cell line looks healthy and it didn't look healthy before, right? Which those experiments are hard. So, yeah.
Okay, thank you.
Okay.